Good morning, students. In this video, let us talk in detail of a DNA structure. In the previous video, we have seen there are two types of nucleic acid called deoxyribonucleic acid, DNA, and a ribose nucleic acid, RNA, where the DNA are abundantly found in the nucleus, whereas RNA are largely found in the cytoplasm of cell. So let's begin with the structure of DNA. So these are the acidic substance present in the nucleus was first identified by a Swiss biochemist Friedrich Miescher in 1869. He was actually investigating the action of pepsin on a pus cell, how this pepsin work in the pus cell, and the undigested residual compound which were obtained from the nuclei of such pus cell showed this acidic substance, and he called it as a nuclein. This happened in 1869. It was again in 1899, Altman, he named it as nucleic acid. Here it is nuclein, but it was Altman who called it as nucleic acid because of its acidic nature. Later on it was subjected to chemical analysis by many scientists, and there are many scientists who has discovered. See, here it was the Fischer. He was the one to identify the presence of nitrogen compounds, that is a purine and pyrimidines, and later on again Kozel, he identified two types of purine, that is adenine and guanine, and two types of pyrimidine, thymine and cytosine in the nucleic acid. For this they were also awarded Nobel Prize in 1910. Then it was Leviny, a Russian biochemist. He recognized this 5-carbon ribose sugar and a phosphoric acids which are present in the nucleus. Like this, the nitrogen bases, pentose sugar, phosphoric acids were discovered, identified, but it was Charkov in the year 1947. They have done this detailed chemical analysis of the DNA, and Wilkin, his full name is Maurice Wilkin, he found out the physical nature of DNA through X-ray diffraction method. This is important. Yes, due to using this X-ray diffraction method, they were able to find the physical nature of the DNA, and based on these data what was given by the Wilkin, it was then the James Watson and Francis Crick, they proposed the double helix model of DNA, and for this they were awarded Nobel Prize, and this was in the year 1953. The double helix model of the DNA was proposed by Watson and Crick, but the reason for this is based on the data which was provided by the Wilkin using this X-ray diffraction method. They have found the physical nature of the DNA. Using this data, they were able to propose this double helix structure of a DNA. So here it will be asked for 5-mark question. What are the salient features of double helix model or Watson and Crick model? They can ask you double helix model or Watson and Crick model. So now the salient features are, they found that the DNA was made up of 2 polynucleotide chain. So DNA is a double-stranded structure. It is having 2 polynucleotide chain. See, let us understand what is polynucleotide chain. I told you polynucleotide meaning when the chain is made up of many nucleotide like this. Now here is a nucleotide joined with another nucleotide. So there are many nucleotides joined with one another to form a chain, and this chain you call it as a polynucleotide chain. And there are actually, this is one chain and there is one more chain So this is one more polynucleotide chain. So that is what they are saying. DNA is made up of, they are made up of two polynucleotide chain. We can find two such polynucleotide chain. Here the backbone is constituted by sugar and phosphates, whereas the bases are projected inside. Meaning, see, if I say nucleotide, what it should have? It should have a nitrogen base, right? These nitrogen base should be connected to a pentose sugar. The pentose sugar are further attached to the phosphate group. One phosphate, this is the nitrogen base, sorry, this is a pentose sugar and this is a nitrogen base. Am I clear? Now this constitute what? One nucleotide. Similarly, here there will be one more nucleotide, meaning again I should write one nitrogen base connected to the sugar and then to a phosphate group. So this is one nucleotide and this is one nucleotide. They are connected with one another by the ester bonds here. You can see here these two, that is the sugar and the phosphate, they form the backbone, whereas the nitrogen base is projecting inside. When I write structure, it will be more clear. And second point is they show anti-parallel polarity. The two chain have anti-parallel polarity. So let us understand what is that anti-parallel polarity. So I am going to write the structure again. This is a pentose sugar. So this is a first position, second position, third position here. In the third position of the pentose sugar, you will have a free OH group. And this is a fourth position and this is a fifth position which is having a phosphate. And at the first position, you have the nitrogen base. The first position of the sugar is attached to the nitrogen base and the third position will have a free OH group and fifth position will have a phosphate group. Similarly, there is one more phosphate pentose sugar which is having first position nitrogen base, and as usual it will also have at the third position, it is having a free OH group, and this will be the fourth, and fifth is again the phosphate. Now here you can see there is a link or a bond formed between the fifth and the free OH group, which is free, meaning it is a reactive. Here you can see a bond. Now this nucleotide is attached to the next nucleotide. Similarly, if I write one more, this is a nitrogen base, OH, fifth position is phosphate. Again you can see the bond here. So this is one chain. The one end of the chain is having a 5' phosphate group. On the other end, it is the 3' OH. This is now free. It will now react with the next nucleotide. But when you take a chain, it will have two ends. The one end of that will have a phosphate group at the fifth position. Therefore, you call that end as a 5' end. Other end, where 3' OH is free, you call it as a 3' end. This is one chain. Similarly, you will have another chain, but the difference here is, here it shows the pentose sugar is this one. Here this is a first position where you will have the nitrogen base. This is a second position, and the third position will have the third position we will have the OH group, and this is the fourth position, and fifth is having the phosphate group. Similarly, take one more Again same, the first position will have a nitrogen base, second, third position will have the OH, fourth and fifth. Now bond is between the free prime OH and the phosphate group. So you can see the bond here. Now nitrogen base, OH, bond is between these two. This was one chain. On the other chain, if you look at the second chain here, this is the 3' OH end, whereas this is a 5' phosphate end. That is simply, I can say, if one chain is from 5 to 3' end, the other chain will be parallel to it. But polarity wise they are anti-parallel, meaning if it has 5 to 3, the other chain will be from 3 to 5. It is running in the opposite direction. If this is one chain, if this is a 5' to 3', another chain is not like this. It is anti-parallel. 5 to 3, here it will be 3 to 5. Like this, they show the anti-parallel polarity. If one chain has polarity 5' to 3', another one will be from 3' to 5'. It will run in the anti-parallel way. Now, third point here is the base in two strands are paired through hydrogen bond. See, you can see here, there is a nitrogen base here and here is one nitrogen base. This is of one strand and this is of the second strand. These two are paired here by means of hydrogen bond. So this here, the bond between two nitrogen base here is the hydrogen bond. Now, DNA are complementary in nature, that is always purine pairs with the pyrimidine. Nitrogen base pair, they show a specificity. See, nitrogen bases are there. So what are the 4 nitrogen bases which are present in the DNA? One it is adenine, guanine and cytosine and thymine. So these are the 4 nitrogen bases present. These base pairing, that is pairing, they show specificity. Specificity meaning it is purine always will pair with the pyrimidine. See, now A is a purine. So if I write A is a purine, it should definitely bind with the pyrimidine on the other side. So what are the pyrimidines present here? That C and T are the pyrimidines here and these two are the purine. So purine always should pair with the pyrimidine. They are so specific that if on one strand adenine is present, on the other side definitely there is a thymine. If this one strand is having thymine, on the other side it will have the adenine. Similarly, if it is guanine, on the other side it will be cytosine. This is called specificity, that always adenine pair with the thymine and cytosine always pairs with the guanine. And there are 2 hydrogen bond between A and T and 3 hydrogen bond between C and G. Am I clear with this point? The base pairs in 2 strands are paired through hydrogen bond, meaning what they are saying, see, this is one strand and this is the another strand. This strand nitrogen bases are these 3. On the other strand you can also find the nitrogen bases. Now there is a bond between these 2 nitrogen base and that is called hydrogen bond. And base pairing is also very specific. Suppose here I have written as nitrogen base. Here if you think it is A, adenine is present. On the opposite side, definitely there should be the thymine present. And Between A and T, there is 2 hydrogen bond. Suppose if in this nitrogen base it is C. On the other side, it should be definitely G. And the hydrogen bond between these two are 3. If it is G here, on the other side it will be C. With the 3 hydrogen bond like this. Am I clear? Adenine always pairs with thymine. Cytosine always pairs with a guanine. And there is 2 hydrogen bond between these two. There is 3 hydrogen bond between these G and C. Right. Now, it's a right-handed fashion. The pitch of the helix is 34 nanometer or 34 angstrom. 10 base pair in 1 turn. To understand this, we need to draw the DNA structure. Yes, practice writing DNA structure. This is not comfortable for me to write. So this is one strand what I am writing. Yes, this is one strand what we are writing. Suppose if this is starting from 5 prime to the other strand is a 3 prime, right? The other strand what we are going to write here, they should be parallel like this. Just come from back of the strand. Now, this is the second strand. The other strand should be from 3 prime to 5 prime. See again, I am going to repeat. It is made up of 2 polynucleotide chain. This is one polynucleotide chain and this is the other one. Therefore, the first point is clear. It is made up of 2 polynucleotide chain. Second one is they show anti-parallel polarity. You can see these 2 strands. The 2 strands are parallel in nature. They never cross each other. If you just imagine a spiral staircase, the 2 railings which are there, they never intersect with each other. They are parallel. So this strand here will be parallel to the other strand. They never intersect. So they are anti-parallel, showing this polarity also you can see. If one strand is starting from 5 prime to 3 prime, the other strand will be from 3 prime to 5 prime. It runs in the opposite direction. The second point is clear. The third point here is, these are the backbone. In the backbone, you can find the sugar and phosphates are present in this backbone. Everywhere you can find the sugar and phosphates and the nitrogen bases which are there here. They are projecting inside. You can see the nitrogen bases here. They are projecting inside, whereas this phosphate group and the sugar will form the backbone. And here also I told you the base pairing had the specificity. That is, if here it is an adenine, on the opposite strand it will be the thymine. And these two will have double bond. If it is C and G, it will have triple bond. Again, if here it is G, it means again here it will be C. And this is triple bond. If it is T, it will be A with the double bond. So like this, you can find the nucleotides arranged. The pith is this. We are talking of this diameter here. It is about 20 angstrom. The diameter is 20 angstrom, and one turn is, how to calculate here is, see, you can find here this one is called as a minor groove. The small depression here, it is called as a minor groove, and this depression, a deeper one here, you call it as a major groove. There's a deeper depression called major groove. Again, you can see one more minor, then it comes the major, right? Now, here the distance from one minor to the next minor, this you call it as one turn. It is called one turn. The distance between one minor groove to the next successive minor groove, it forms the one turn. That is the 360-degree turn, you call it as one turn. Suppose here is one staircase step. You have to cross like this. We are just crossing and we'll come back to the step which is upper to this thing. You call it as a 360 rotation or 361. This forms one turn. In one turn, there are 10 base pair present. In this case, I have written 1, 2, 3, 4, 5 base pair I have shown. Watson and Crick model say that in one turn, here the one turn I told you it is 34 angstrom, and in one turn we have 10 base pair. So now if you want to calculate what is the distance between the successive base pair, am I clear? 1, 2, 3 like this, there are actually 4, 5, 6, 7, 8, 9, 10. 10 base pair will form one turn. If the length from 1 to 10 is 34 angstrom, what is the distance between these base pair, the first base pair and second base pair? Just divide it by 10. That is 3.4 angstrom. Otherwise, you can also say it in terms of 3.4 nanometer. 3.4 nanometer. If it is in the nanometer, then it is divided by 10. That is 0.34 nanometer. Am I clear? Either say in terms of angstrom, 34 angstrom or 3.4 nanometer. The distance will be 0.34 nanometer or 3.4 angstrom. See, coming back to this, the pith of the helix, that is one turn, is 3.4 or 34 angstrom. There are 10 base pair in one turn. Therefore, the distance between the successive will be, just divide this by 10. The length of the DNA is always measured by number of nucleotides. See, this is extra point. This much you have to explain. If they ask you for Watson and Crick model, make these points very clear and practice along with the diagram. Right? Now, the length of a DNA is always measured by number of nucleotides depending on how many nucleotides we have. For example, if you take a bacteriophage, that's a lambda bacteriophage, the total number of base pair what it is having is 48,502 base pair. It is the length. The DNA of this bacteriophage has 48,502 base pair. If you take in case of humans have 3.3 into 10 to the power of 9 base pair. That is 10 raised to 9. Just multiply. So many base pairs are present in the human DNA. If you consider the E. coli, which is a bacteria, this shows 4.6 into 10 to the power of 6 base pair. So the length of a DNA can be measured. More the number of nucleotide, the length of the DNA increases. Very simple. Am I clear? the amount of DNA per nucleus of a species is measured in terms of picograms. All these are extra, don't worry, but remember these points, the salient features of Watson and Crick or double helix model. It is made up of two polynucleotide chain. They are anti-parallel polarity. If one runs from 5' to 3', another will go from 3' to 5'. The base pairs here between the two strands are paired through hydrogen bond. There are 2 hydrogen bond between A and T. There are 3 hydrogen bond between C and G. Always adenine pairs with the thymine and thymine with the adenine. Cytosine always pairs with the guanine and guanine always pairs with the cytosine. Here, if adenine is a purine, thymine is a pyrimidine. Again, here we know this is a purine. This is a pyrimidine, and guanine is a purine. Purine always pairs with the pyrimidine. Pyrimidine always pairs with the purine. And it's a right-handed fashion. And the coiling is right-handed fashion, and one turn is measured as 34 angstrom. There are 10 base pairs in one turn. How to calculate one turn? The distance from one minor groove to another minor groove, or you can calculate from one major groove to the next major groove. That will be form the second turn, like that. Now, there are types of DNA on the basis of function. There are tropic DNA and genetic DNA. Tropic DNA are all activities. They are responsible for all activities except the reproduction. All the metabolic reaction, whatever it happens, they are responsible for all such activities except the reproduction. But genetic DNA are inherited, that they are responsible for reproduction. This is if DNA is classified on the basis of their function. The more important here is the DNA classification on basis of the number of base pairs present in one turn per turn. The number of base pair per turn. On basis of this, the DNA are classified into A DNA, you call it as A DNA, B DNA, C DNA, D DNA and Z DNA. You can see here, in case of A DNA, the number of base pair present in one turn is 11. But what we discussed previously, that was Watson and Crick proposed, that was the B DNA. That is why in one turn we have found 10 base pair. Again, cDNA 9.3. 9.3 meaning what? Between 9 or 10. If in one turn, if the number of base pair are between 9 and 10, it is called cDNA. If it is 8, it is dDNA. It is 12, maximum is 12, that is zDNA. And here all of them except zDNA, they are right-handed. They are coiled right-handed. But left-handed is seen in case of zDNA. Clear? Very important. Watson and Crick proposed whatever features we have discussed previously. That's a Watson and Crick model. It was regarding the pDNA. And they can ask you for zDNA because it is different, because number of base pair is also more, and it is the only left-handed DNA, whereas all other are right-handed DNA. There is one more rule which is called as Charkov rule. It is Erwin Charkov. He has proposed a rule. So what it says is, always the number of purine is equal to number of pyrimidine. Very simple. If you consider a DNA, suppose in a DNA the number of purine is 20. What will be the number of pyrimidine? According to Charkov rule, it says that in a DNA, number of purine is always equal to number of pyrimidine. If the number of purine is 20, the number of pyrimidine will also be 20. Very simple, because we know that purine always pairs with the pyrimidine. Clear? So, and remember, this is applicable for double stranded. This is applicable for double strand, when DNA is a double strand, not for any single strand DNA. There can be questions asked, like if the number of adenine is 20, adenine is 20, then just by knowing the number of adenine, you can calculate the remaining base pair. How? We know that adenine will pair with the thymine. So if adenine is 20, what is the number of thymine present? Thymine will also be 20. Clear? If adenine is 20, thymine will be 20. Now A plus T is how many? 40. Remaining is how much? Out of 100, 40 is given. Therefore, remaining is only 60. In 60, the cytosol will be 30 and guanine will be 30. This is how you can calculate. If one base pair is known, you can calculate the number of other base pairs which are present. This is all possible only because the DNA are complementary in nature. If the one of the strand, I told you 2 strands are present. If DNA meaning, we should write 2 strand here. So if this strand is having the base pair like this, this is a nitrogen bases of one strand. By knowing this, you can write the base pairs of the second strand. How? Because A always pairs with the T, T with A with T, C with G, G with C, C with G, T, A, T. Like this. By, if you get one sequence, you can calculate the other sequence, other strand sequence. Am I clear? This is very important. It will be asked for 5 marks. Just go through this salient features.